You're much welcome again in this session where we are talking about cell-mediated immunity. In the first part, we have introduced major histocompatibility complex, we have seen cells involved, and we have seen exogenous pathway. So in this session, we want to focus on the endogenous, or what we call cytosolic. You can call it endogenous or cytosolic pathway. And this cytosolic or endogenous pathway is responsible for, we saw in the introductory part that the major histocompatibility class involved is major histocompatibility type 1, which presents intracellular pathogens or tumor cells or intracellular bacteria. And we saw that T cytotoxic is the one that is involved, whereby in the previous we saw T helper cell is the one which is involved. And we saw that also this major histocompatibility complex type 1 is presented by all nucleated cells. So all nucleated cells within the body do this role of fighting against intracellular microorganisms or pathogens. So as we begin, we remind you to like the video if you enjoy. So this one occurs for intracellular. So we have here intracellular virus. It can be an intracellular virus. It can be an intracellular bacteria, or it can be a tumor cell. So whenever there is a tumor cell or a virus within a nucleated cell, like leukocytes or white blood cells, what happens, this virus or the tumor is processed by triggering what we call ubiquitin. There is a protein called ubiquitin, and this ubiquitin utilizes energy in form of ATP, which activates proteases. This one in form of ATP plus ubiquitin, it triggers proteases, and these protease enzymes, they form what we call a proteasome. And this proteasome is the one that comes and processes this intracellular virus. As it is passing through this proteasome, it is cut into fragments. It is cut into fragments by the action of proteases. These are proteases within the, so the proteases are within the proteasome. So the proteasome contains proteases which are responsible for cutting the big virus into fragments, or the tumor cell into small fragments, which can be presented on the surface. And remember, in exogenous we saw invariant chain or the clip, but this one does not involve invariant chain. So what happens is, on the rough endoplasmic reticulum, after manufacturing this major histocompatibility class type 1, there is a transporter called TAP. TAP is known as transporter antigen protein. Transporter antigen protein. And this TAP is the one that enables entry of these fragments cut. These fragments from the virus, which are cut, enter into the rough endoplasmic reticulum via TAP, what we call transporter antigen protein, and after entering, they come and bind on the major histocompatibility class type 1. And after binding, this class type 1 moves via Golgi apparatus, and is taken to the surface. As we said, this one does not involve invariant chain, because that's why we are taking the peptides within the rough endoplasmic reticulum. So after being transported on the surface, we find this major histocompatibility class 1 is going to present the portion of this virus on the surface. After presenting it on the surface, it presents it to the TCR arm of the T cytotoxic cell. So this T cytotoxic cell has the TCR arm, what you call T cell receptor. And after this T cell receptor binding on the peptide, viral peptide or tumor peptide presented, being enhanced, this binding is enhanced by a CD8 positive arm. And this is a CD8, and that's why we call T cytotoxic cells to be CD8 positive cells, because they have the CD8 arm, which acts as a co-stimulation. So after binding, causing firm attachment or interaction between the peptide presented by class 1 major histocompatibility, it will trigger the T cytotoxic cell to release interleukins, especially like interleukin 2. It will also produce guys like perforins, produces perforins. It will produce also glanzymes, whereby the perforins which are produced by T cytotoxic, in the beginning we told you T cytotoxic kills the infected cell. So it will produce these perforins, and these perforins will come and create pores. They come and start creating pores on the nucleated cell, infected nucleated cell. This is the infected nucleated cell, and this infected nucleated cell is the one that these perforins come and create pores. Perforins come and start creating many pores. They create pores in it. And after creating pores, the glanzymes come and enter. So we see glanzymes passing through these pores created by the perforins. We see glanzymes entering, and after entry of glanzymes will go and activate apoptosis. They are the one that will activate apoptosis, which is programmed cell death. So it will make this cell to undergo apoptosis, and then it is cleared from the saturation. So this is how T cytotoxic cells act, whereas the cytokines, in case the ones which are produced, can also go and also activate the B cells in case the humoral immunity is required. But here we do not require any humoral immunity. So we see cytotoxic T cells producing directly perforins and glanzymes. Maybe the cytokines which are produced, especially interleukin 1, interleukin 2, also can go and activate macrophages and natural killer cells. And these ones can also come and kill this virus or tumor infected cell. So this is what occurs under endogenous, or what we call cytosolic pathway, where we have looked at how it involves the breakage of the intracellular virus or tumor within the cell. And when this virus is detected by the immune system, it activates the protein known as ubiquitin, which in the presence of ATP activates proteases within the proteasome. And these proteases cuts the virus into smaller peptides, which are released within the cytosol, and they enter into rough endoplasmic reticulum, where class 1 is, via transporter antigen protein. And this, it binds. After binding, it moves by exocytosis via the Golgi apparatus and presents a peptide or a portion to CD8, what we call cytotoxic T cells. And after presenting it, it becomes activated and it releases perforins and granzymes. Perforins create pores in the infected cell, and the granzymes come and activate apoptosis, which is programmed cell death. Another thing, interleukin-2 can be released, which activates macrophages and natural killer cells, which can also come and kill the infected cell. Thank you so much for learning with us cell-mediated immunity, where we have discussed endogenous, or what we call cytosolic pathway for majorly viral infected cells, tumor cells, and cells with intracellular bacteria. Thank you so much.